Okay, so in the last video, we talked about what happens when DNA polymerase accidentally incorporates the incorrect base and it's not repaired by the proofreading activity. So we learned about the mismatch repair pathway that's able to recognize mismatches between daughter and parental strand. And then it's able to specifically cleave the daughter strand and repair that side based on the fact that the parental strand is methylated, but the daughter strand is not methylated. So that enables it to tell which one is the correct template to use for fixing the error. But of course, there are many other types of damages that can occur when to the genome that are not a product of polymerase errors. So these can be chemical damage that occurs through this kind of laundry list of chemical processes that can damage our genome. And in the next two videos, we're going to learn about how what these chemical modifications are and how the cell goes about fixing them. And so why are these chemical catastrophes potentially catastrophes, right? I mean, couldn't these chemical modifications have no effect on base pairing? Well, that's always a possibility, but remember that the genome integrity, that is this idea of being able to have a perfect genetic material that can be used as a template to copy perfectly and pass on to the next generation, really depends on the specificity of base pairing. And so we talked a lot in the previous lectures about how A is only going to pair with T and C is only going to pair with G. And that perfect orthogonality between those base pairing interactions makes it so that when you unwind the two strands, it's trivial to completely and accurately copy that strand via complementary synthesis. However, these base pairs and these base pairing interactions that are mediated by nucleobases, well, those are just chemical compounds, and the interactions that dictate their pairing is just chemistry. And so if chemistry happens to the nucleobases, then it can affect the types of interactions that can occur, and that might change the types of base pairing possibilities that are out there. And so if the nucleobases were to become chemically modified, and the hydrogen bonding that sets AT or CG base pairing gets disrupted, then this can lead to incorporation of the wrong base during DNA replication. And this would result in a mutation being passed down to the next generation. So instead of inheriting a perfect copy of the parental genome, the daughter cell would inherit a copy that contained changes, inaccuracies relative to the parental strand. And so what you should imagine here is if we had some parental strand that we were copying and there was some chemically modified base here, so this guanine was now modified in some way, and that modification caused a T to be preferentially added instead of a C, then the daughter strand is going to inherit a T in a position where it should have had a C. And so this is exactly the type of phenomena that we have to worry about if we're a cell. What we're going to do now is go through the different ways in which these chemical modifications can occur that can lead to these problems. So what are some chemical modifications that could disrupt base pairing interactions? So we have this perfect adenine nucleotide monophosphate here, and what types of chemistry can we do to it, and what types of chemistry can disrupt base pairing interactions? So one of the common things that can happen to a base like this is a process called alkylation. And alkylation is the act of adding methyl or ethyl groups, or really any sort of alkyl group, to the nucleobase. And so you see here, for example, at this nitrogen position here, normally there is a lone pair, but following alkylation, we have deposited this methyl group. And in fact, this base is now positively charged, but that's not always the case. In general, you should just think of alkylation as adding methyl or ethyl groups to the base. And if we compared these two nucleobases that you see here, and we looked at what happened when you try to base pair with thymine, the intended complement to adenine, recall that in the native interaction, we have donor and acceptor here, pairing with acceptor and donor here on our thymine. It looks perfect. A and T, 2 hydrogen bonds. But in the case of the alkylated adenine, we now have lost our hydrogen bond acceptor here, right? And so there's nothing for this donor to pair with. And so we're left with only 1 hydrogen bond here, and that's really not going to be enough to give us a strong base pairing interaction. And so if the alkylation of our nucleobase occurs at a position involved in canonical base pairing interactions, then there's a really good chance that it's going to disrupt the ability to form those hydrogen bonds and thus disrupt the base pairing. However, there are other times in which the methylation occurs at a site that's not involved in base pairing. And in fact, in the previous lecture, we talked about methylation of adenine in mismatch repair. And those modifications occur at places that don't affect the ability of the base to form base pairing interactions. And so here's an example of just such an alkylation event here, where by putting a methyl group at this nitrogen here, because this nitrogen is not involved in canonical base pairing interactions, it's a safe site of alkylation. So you could put a methyl group here and you'd still expect to have your donor and acceptor groups available to form hydrogen bonds. And so that's something important to think about with all of the chemical modifications that we're talking about, is sometimes they are going to occur at locations that are very disruptive to base pairing, while other times they're going to occur at locations that's benign. And as long as you know which sites on your nucleobases are involved in hydrogen bonding, then you're going to be in good shape. So what are some other chemical modifications that can occur that can disrupt these base pairing interactions? So something that occurs spontaneously from time to time to these nucleobases is a process called deamination. And as its name would suggest, deamination is going to replace an amine, hence D-amine deamination, will replace the amine with a carbonyl group. And so here you have our adenine undergoing a process of deamination, where now the amine has been converted into a carbonyl group here. And as before, you will notice that we have changed the situation from having hydrogen bond donor acceptor at these positions here to now being acceptor donor. So that's not good. This type of deamination here is going to change those H bond donor acceptors and likely disrupt hydrogen bonding. And in fact, most of the nucleobases that you commonly observe can undergo deamination to bases that you may or may not have heard of. They often have more exotic names. So when adenine undergoes deamination, it becomes hypoxanthine. When guanine undergoes deamination, it becomes xanthine. And interestingly, when cytosine undergoes deamination, it becomes uracil. And that's kind of a sneaky one, because unlike hypoxanthine and xanthine, where these are not canonical bases that are used for anything, uracil is something that comes up often in terms of base pairing interactions. It's not native in DNA, but we do know that uracil can make base pairing interactions with adenine, and so this is a particularly devious one that may impact reading in a very obvious way. And of course, it's important to note that you can't undergo deamination if you don't have any amines with which to do that process. So bases like thymine or uracil itself, which do not have amines, cannot undergo deamination. So just keep in mind that every base sort of has its corresponding, with the exception of thymine and uracil, has its kind of corresponding deaminated form. And as before, these deaminated forms, because you've changed the donor and acceptor patterns on them, can create problems for base pairing interactions. So again, a native AT, we've got donor acceptor making interactions with acceptor and donor on thymine. And in the deaminated form here, we will see that we have actually two problems. Basically we have two acceptors and two donors, so there should also be an X here. I apologize for that. I'll fix that in the notes here. But basically you can't form the hydrogen bonding interactions here at all. And so we've completely disrupted the ability to form these interactions. And interestingly, not only do you disrupt the native interactions, but there are alternative base pairing interactions that then can become favorable. So if we have deaminated cytosine down to uracil, then actually it is very content to make a base pairing interaction with adenine, right? And this is actually the native way that these would pair in RNA. But now we've converted what would normally be a CG base pair into something where we would like to get a UA base pair. And then similarly, when you have an exotic base that's like hypoxanthine that's produced from deamination of adenine, now our hydrogen bonding pattern is such that, again, we can form, instead of being able to form base pairing interactions with thymine, we can form them with cytosine. And so what should have been an AT pair is now going to be an AC pair. And so these alternative base interactions are what can lead to misincorporation of nucleotides if we try to copy strands that contain these deaminated residues. Okay, last type of chemical modification that can be disruptive occur from a process called depurination. And so remember that there is a glycosidic linkage between our sugar in the DNA, our deoxyribose here, and the nucleobase. So we have this N-glycosidic bond here. And if this glycosidic bond were to be broken, we would say that the base has undergone a process called depurination in this case, which leads to release of the purine base from the nucleotide. And we are left with basically a sugar that has no, a sugar phosphate here that has no nucleobase on it. And good luck getting this thing to base pair with anything because it doesn't have any base at all. So it's basically just kind of like a naked empty space. And that's going to again create problems for the replication process. Okay, last type of chemical modification that we have to discuss is a little bit more complicated because it doesn't occur to a single base, but it occurs to adjacent bases in the genome. And so if you have adjacent thymines or cytosines, so if you have adjacent pyrimidines in your genome, then in the presence of UV light, those pyrimidines can become cross-linked together. So there's some interesting photochemistry that we're not going to get into that can cause the two pyrimidines to become cross-linked into what is called a UV dimer, a pyrimidine dimer or a UV dimer. Okay, and these pyrimidine dimers introduce a very bulged and distorted DNA structure that you can see here. So here's an actual pyrimidine dimer in a structure of DNA. You can see this has introduced sort of a kink or bulge in the backbone here. And this abnormal structure is very difficult for DNA polymerase to correctly read and process. And so you're often going to get the incorrect bases added if you try to copy by these thymine dimers here, okay? And so basically, to sum up all the types of chemical modifications here, basically if you can just draw a picture of a nucleotide and explain to yourself what alkylation is, what deamination is, what depurination is, as well as know about these UV-induced dimers that can occur with pyrimidines, then you're going to have a pretty good handle on the types of damage that can happen to these nucleotides. And you have a good handle on whether or not a particular chemical modification to the DNA is going to be problematic for base pairing. And so the key idea here is to know your damage and to know whether when a site becomes damaged, if that's going to have consequences or not for base pairing. And obviously we discussed scenarios for each one of these in which, yes, it is a big problem. So once you know all those damages though, boy, I mean, it seems like your genome is probably a disaster. But the good news is that as long as the cell fixes all of these chemical modifications before it tries to initiate DNA replication, then things should be okay, because it's really only the replication process that's going to lock in the problems associated with this damage. And so in the next video, we're going to talk about how for each one of these types of chemical damage that we've talked about, how the cell can fix that damage to make sure that replication goes smoothly. And so you'll learn that in the next video.